Short questions with their answers. Proteins overview. 1. What are the critical roles of proteins in the body? Proteins are required for structure, functioning, maintenance, and regulation of body systems. 2. What elements are commonly found in proteins? Carbon, hydrogen, oxygen, and nitrogen. 3. Name 2 additional elements that may be present in some proteins. Phosphorus and sulfur. 4. What trace elements may be incorporated into proteins? Iron, iodine, and magnesium. Structure of proteins. 5. Define proteins chemically. Proteins are polymers of amino acids or polypeptide chains. 6. What are the 2 types of protein structures based on polypeptides? Single polypeptide and multiple polypeptides. Amino acids. 7. What are the building blocks of proteins? Amino acids. 8. How many amino acids are commonly found in proteins? 20 amino acids. 9. What determines the chemical properties of amino acids? The R group. 10. What groups are attached to the alpha carbon in an amino acid? A hydrogen atom, an amino group, a carboxyl group, and an R group. Essential and non-essential amino acids. 11. What are essential amino acids? Amino acids that cannot be synthesized by the body and must be obtained from the diet. 12. Why are essential amino acids important in the diet? They are needed for building and repairing muscles and bones, and for synthesizing hormones and neurotransmitters. 13. Name the 9 essential amino acids. Lysine, leucine, isoleucine, histidine, methionine, phenylalanine, threonine, valine, and tryptophan. 14. What are non-essential amino acids? Amino acids that can be synthesized in the human body. 15. Name the 11 non-essential amino acids. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, and tyrosine. Peptide bonds. 16. What is a peptide bond? A bond formed between the carboxyl group of one amino acid and the amino group of another amino acid. 17. How is a peptide bond formed? By the condensation of 2 amino acids, releasing a water molecule. 18. What happens during protein hydrolysis? Peptide bonds are broken, releasing individual amino acids. 19. What is the role of peptide bonds in protein structure? They maintain the primary structure of proteins. 20. What process forms dipeptides and polypeptides in cells? Translation on ribosomes. Structural conformations in proteins. 21. What is the primary structure of a protein? A linear sequence of amino acids in a polypeptide chain. 22. What bonds help form the secondary structure of proteins? Hydrogen bonds. 23. What is the tertiary structure of proteins? A 3-dimensional globular structure formed by folding of the alpha helix. 24. What is the quaternary structure of proteins? A complex structure where more than one globular structure interacts. Amino acid sequence significance. 25. What determines the sequence of amino acids in a protein? The sequence of nucleotides in DNA. 26. What is the effect of a point mutation on a protein? It can change the amino acid sequence, causing defects like sickle cell anemia. 27. What change occurs in hemoglobin during sickle cell anemia? Glutamic acid is replaced by valine at position 6 in the beta chain. Classification of proteins. 28. Name the 2 types of proteins based on structure. Fibrous proteins and globular proteins. 29. Give examples of fibrous proteins. Collagen, fibrinogen, actin, myosin, and keratin. 30. Provide examples of globular proteins. Enzymes, hormones, antibodies, and channel proteins. Roles of proteins. 31. What is the role of collagen? It forms the matrix of bones, cartilages, and components of tendons and ligaments. 32. What is the function of keratin? It strengthens protective coverings like hair and nails. 33. How do enzymes function in the body? They speed up biochemical reactions. 34. What is the role of hemoglobin? It transports oxygen and carbon dioxide. 35. What is the function of fibrinogen? It helps in blood clotting. 36. Name 2 storage proteins and their sources. Ovalbumin, egg whites, and casein, milk. Biochemical tests for proteins. 37. Describe the Biuret test for proteins. Add NaOH and copper sulfate to the test solution. A violet color indicates the presence of protein. 38. What is observed in Millon's test if protein is present? A white precipitate forms, turning red upon heating. 39. What does the Ninhydrin test detect? The presence of amino acids or proteins.